Okay, our topic is HIV/AIDS and other retroviruses. Okay, before we go to the lecture proper, we are presented here with a case of a 28-year-old male call center agent from Makati who came in due to difficulty of breathing. He had a 3-day history of cough that is associated with low-grade fever. Physical exam showed normal except for this oxygen saturation. The normal is 95 and above. Pero our patient had an 88% cyanotic, meaning there are discoloration sa lips, sa nails, meron ding retractions or cervical lymphadenopathies, mga kulani, and maraming breath sounds niya, yung pag humihinga siya maraming phlegm. Other findings were unremarkable. Now, for this kind of case, very timely to say that we can suspect HIV or AIDS. All right. Now, when you say AIDS or it is caused by a human immunodeficiency virus or HIV that specifically attack CD4 T lymphocytes. Okay. The last stage of HIV infection is actually the AIDS, and the HIV belongs to the family of retroviruses, now which contains 2 copies of single-stranded RNA with a genome that is packed with several enzymes. Okay. Facts, according to the Department of Health, last June of 2021, there are 24 new individuals diagnosed HIV in the Philippines every day of March 2017. That's the highest number of cases that is ever reported since 1984. Okay. Last June of 2021, 88,108 just from January 1984 to June of last month of this year, 88,000. The top 5 regions would include the NCR being on top, regions 4A, 3, 7, and 6. For the population that is most commonly affected or infected, 84% would be coming from a sexual contact, male to male interaction. 10% would be from bisexual. Male, predominating the data of HIV diagnosis amounting to 5,700 plus, as opposed to female who only tallied 289. 50% of cases of HIV diagnosis aged 25 to 34 years old. Then, second would be 35 to 49 years old. So we can say that the population, the average age of HIV diagnosis every day in the Philippines belongs to the working class or your working group. Okay, HIV. No, it binds to CD4 cells. That's the target cell ng HIV, and it becomes internalized. The virus replicates itself by generating a DNA copy, but this is through reverse transcriptase. Okay, viral DNA becomes incorporated into the host DNA. That is replication. So if it is DNA to DNA, it's replication. But if it is an RNA who wants to generate a DNA, kabaliktaran, reverse transcriptase. That's the enzyme involved in that process. HIV is a member of retrovirus and lentivirus, and it cannot multiply on its own. It reduces the number of CD4 lymphocyte or CD4 cells in our body, making the person more likely to get other infections. CD4 cell class, or the CD4 plus lymphocyte, it's actually a cell of our immune system. It helps us in competing with the viral infections, bacterial infections, but in HIV, it reduces its number. That is the cell that is the target of your HIV virus. It can destroy so many of these cells that the body can't fight infection or opportunistic diseases. You just look at it as if you have the HIV, your immune system is attacking the HIV. Now, once you have infection, another infection, your immune system will shift its fighting mechanism or immune system will shift to the new infection. Thus, the HIV now will be multiply again. Okay, HIV infection based on this slide is a lifetime disease. Okay, it carries only 2 single-stranded RNA and it involves reverse transcriptase enzyme. Meaning, as mentioned, if you want to convert RNA to DNA, you need this enzyme reverse transcriptase. Okay, when you want to replicate DNA to its DNA again, it's called replication. If it is DNA to RNA, that term is your transcription. But in HIV here, you want to replicate DNA using your RNA template. So RNA to DNA, the process is reverse transcription. And if you want to have an RNA to be converted to a protein, that's what we call translation. This is the basic biochemistry of nucleic acid. Okay, so HIV genome. This is a little complicated. No, just listen. HIV genome encodes for 9 genes. You have the structural proteins, regulatory proteins, and accessory or auxiliary proteins. This is the HIV structure with so many complex enzymes, okay, or genes. Okay, this is the HIV structure. As you can see, no, we have this P. Yan mga yan. These are your GAG genes. Your P55 is actually the precursor of your GAG genes. P17, that's the matrix. No, this is the matrix of the HIV that helps hold the envelope or the nuclear envelope proteins to the virus itself. The P24 here, as it indicated by this blue color, that is your capsid, and your P7 inside is the nucleocapsid. These are your GAG genes, your P55, P17, that's the matrix, your capsid, P24, and your P7 as your nucleocapsid. For the Pol genes, you have reverse transcriptase. That's your P66 or P55, your protease or your P10, and your integrase as your P31, as exemplified by these labels. Your reverse transcriptase enzyme, that is your P66 or P55. It copies the viral DNA to become, it copies the viral RNA to become DNA. As the name implies, reverse transcription using a reverse transcriptase enzyme. Your protease, that's your P10, it processes proteins that is made from HIV's genome so that they can become part of the newly functioning HIV particles. Another enzyme is our integrase, that's your P31. It integrates the DNA copy of HIV's genome into the host DNA. So basically, these Pol genes are enzymes involved in the viral multiplication and integration of the DNA virus to the DNA host. And lastly, the end genes or the envelope genes. These are exemplified by the following: GP120, GP41. Those are your envelope genes. Your GP160 is the precursor of envelope glycoproteins. While look at the GP120 here. This helps the virus attach to the CD4 of the host cell or the host receptor. These are that will serve as the attachment of that virus to the CD4 cell, the host cell. So this is the viral structure, and GP41 is actually, it helps the virus fuse within the cell. So GP120 attachment, GP41 helps the virus to fuse with the cell membrane and enter the cell. So this one is for fusion, GP120 is for attachment. So those are your end genes or envelope genes. Okay, this is the summary of the structure of the HIV. The GAG genes, they are known as for a genomic region encoding the capsid proteins. As mentioned, these are the GAG genes. Your POL, these are your enzymes, a genomic region that encodes the viral enzymes such as reverse transcriptase, protease, and integrase. And ENV genes, these are genomic region encoding for glycoprotein, kaya siya GP. It serves as your glycoprotein 160, 120, and GP41. 120 as a review, this for attachment, while GP41 is for the fusion. Okay, structural proteins. These are the different. Now we have the GAG, the POL, and the ENV genes. Now we can also discuss this gene based on its function. The GAG genes are the group-specific antigen. No, group-specific antigen in its function as exemplified kanina sa lecture, core proteins and matrix protein. The POL or the polymerase genes and envelope genes are your glycoproteins. Now look at this structure. These are your different genes. What about the regulatory proteins? There's a quite few regulatory proteins. These are the TAT or the transactivator here. They are positive regulator of transcription. It accelerates the production of more HIV virus. And the rev genes, just like above the envelope gene and TAT genes, they are the regulator of your viral expression. They will stimulate the production of HIV proteins, but it suppresses the expression of HIV's regulatory gene. So they are the rev, the regulator of viral expression, and your transactivator. These two genes are your regulatory proteins. Next, what are the accessory proteins? Based from this viral structure, these are the following accessory proteins. We have the NEF or the negative replication factor. It retards the HIV replication. The VIF or the variant ineffectivity factor. It increases the ineffectivity of the HIV. Your viral protein R transport the DNA to nucleus. It also accelerates the production of HIV proteins. The viral protein U, no, viral protein U, it promotes intracellular degradation of that CD4 cell. And lastly, your LTR here and here. These are the long terminal repeat. They will act as promoters or enhancers. Ito yung sticky ends which integrates the enzyme protein uses to insert the HIV genome into the gene. Dalawa yan kasi those are their sticky ends. Their LTR are your long terminal repeat. So there you have it. You have your regulatory proteins, your accessory proteins. Okay. So again, these are the complex enzymatic reactions or enzymes found in the HIV. Okay, so please take note of those. Okay, so after discussing the viral structure of the HIV, let's go to the types of human immunodeficiency virus. And there are two. We have the HIV-1 and HIV-2. Your HIV-1 has three distinct groups based on the envelope gene, the glycoprotein. You have the M, N, and O. They contain the genes that encodes the structural proteins of the virus. As mentioned a while ago, these are your GAG, your POL, and your ENV genes. It also contains 6 other genes, as mentioned a while ago. Okay, so these are HIV-1, 3, M, and O. For HIV-2, there are 5 subtypes identified. It is A, B, C, D, and E. No? The major difference with HIV-1 is that, number 1, they lack the VPU gene. Okay, in HIV-2. And they have VPX gene that is not contained in HIV-1. So please take note, in HIV-2, they don't have VPU, but they have VPX gene. Okay, epidemiology, some would say the origin of AIDS in humans happened in Africa. Okay, the first HIV case happened in Cameroon. Okay, in South Africa, in Africa. Cross-species infection by simian viruses, meaning from monkey. Current evidence would be a primate counterparts of both HIV-1 and HIV-2 that is transmitted to humans on multiple different occasions. So there is a cross-genesis of this simian or the chimpanzees, that's a zoonotic type of infection, to become infecting the humans. And in 1930, in 1930, there is an introduction of this simian immunodeficiency virus to humans that give rise to HIV-1 group M. Okay, HIV-1 group M. If you would notice this SV40, this was discussed 2 weeks ago. If it cross-genesis, it cross-reacts with through the zoonotic infection, it becomes HIV-1. Okay, that is very indigenous to Western Africa. Okay. So the human immunodeficiency virus, they are transmitted through the following. Okay. They are transmitted through sexual contact, by passage of your semen and vaginal secretions, needle sharing through blood, no injection of your for those drug IV abusers, mother to child transmission. There are presence of HIV in the breast milk, and for contaminated blood products or blood transfusion products and organ transplantation. So these are the manners of transmission of HIV. You cannot get HIV from this just by kissing, hugging, sharing food, insect bites, sweat, sneezes and coughs, bathing, and toilet seats. Actually, ito, majority COVID-19, not HIV, right? But you cannot get HIV from these manners of transmission. Okay, as you all know, HIV can be transmitted by the following, right? They cannot be transmitted by just using toilet seats, kissing, sharing cutleries, and just by touching. Okay? All right, high titers of HIV virus, they are found in the following: blood, semen, also in vaginal secretions, but high titers are found in these two specimens, blood and semen. No evidence of viral transmission through a casual contact or kissing, holding, or sharing food utensils. Okay. Before the HIV affects the CD4 cell, what happens is that during the sexual contact in your vagina, there are macrophages and dendritic cells, right? So it goes to the lymphatic system and eventually it goes to the blood. Same with sexual contact in men. No, it goes to the seminal fluid, goes to the lymphatic system, and it goes to our blood. So after discussing the epidemiology, the manners of transmission of HIV, the types of HIV, let's go on to the life cycle. Okay, life cycle would include the following. No, it's not as complex as what you think. No, if you would just follow the slide that I gave you, ayan, dyan lang tayo kukuha ng examinations. Okay? The life cycle of HIV begins in binding, infusion, reverse transcription, integration, replication, assembly, and budding. This happens in the CD4 cell of the host. Okay, so this is the HIV virus. There is attachment and fusion. This will be discussed in a while in the succeeding slide. Okay, step 1, binding. So in binding, the HIV virus would attach itself to the receptors of the surface of the CD4 cell. So these are the CD4 cell. There is CCR5 co-receptor, CD4 receptor, and CXCR4 co-receptor. So meaning, in the CD4 cell, they have this receptor to the HIV. Okay, that's binding. Next would be the fusion. In fusion, if this is the HIV envelope, Okay, this is the CD4 cell membrane. The HIV envelope and the CD4 cell membrane fuses together, which allows entry to the CD4 cell. Okay, this one will allow entry to the CD4 cell after it attaches to the CD4 cell. So now the virus fused now with the CD4 cell of the host. What would happen inside the CD4 cell? It undergoes HIV reverse transcription using its enzyme. So HIV RNA, we all know this. By reverse transcription, we use reverse transcriptase to become HIV DNA. This will be incorporated now to the CD4 cell. Now, to the CD4 cell of the host, pupunta yan sa nucleus now, and that will start its conversion of its genetic material from RNA to DNA by this reverse transcription. So again, dito pa lang siya before siya pumasok sa nucleus. Okay? Next step would be integrase. In the integration or integrase, the integrases inside the CD4 cell, the HIV virus, ito, releases the so-called enzyme integrase. This is an HIV enzyme. It will integrate to insert to its viral DNA, to the DNA of the CD4 cell. So this one is actually, the blue ones are the CD4 cell DNA. What would happen is that in this integrase enzyme of the HIV, this is the HIV virus or DNA. It will integrate now to the CD4 cells DNA. Okay, this happens in the nucleus. So in the integrase step happens in the nucleus, while in the sa labas pa lang, reverse transcription, while in the cell membrane of CD4, attachment and fusion happens. Okay, what would be the next step after integrase or integration of this HIV DNA to the host cell DNA? There will be replication. Okay? In replication, once integrated, the HIV now begins to use machinery of the CD4 cell to make a long chains of HIV proteins. Okay? By making long chains of HIV proteins, the proteins now the protein chains are actually the building blocks for more HIV virus for replication. Okay, that would be called replication. What is the next step? If there after the replication, assembly step would be the next step. No, the new HIV and the HIV RNA now will move to the surface of the cell, and they will assemble here into immature non-infectious HIV. So after replication, this HIV proteins and RNA will now move towards the cell membrane. Okay, they will be tagged as immature non-infectious. Okay, at the assembly. What about the budding stage? The step 7 would be the budding stage. Newly formed immature, which is the non-infectious, will be pushes itself out of the host CD4. Okay. The new HIV will release protease. Again, that's an HIV enzyme, and that will act to break up the long protein chains that will form the immature virus. Yung smaller HIV proteins will combine to form the mature or infectious HIV. So here, this is the protease enzyme. That's an HIV enzyme that is very crucial in converting the immature HIV proteins and DNA to become immature infectious ones. Okay, that's based on the protein protease. There. So after the products of mature infectious HIV, it will now then infect so many cells, CD4 cells. That's why in HIV patient, for the AIDS patient, the CD4 count starts to decrease, while the HIV virus starts to increase. I'll show you a table showing that graph, no, the CD4 and HIV virus. Okay. So basically, those are the stages of AIDS from attachment to fusion to replication, or from uh, reverse transcription, integration, no, replication. What else? Assembly and budding. Please take note of each step in the life cycle of an HIV. Okay. All right. So after discussing the life cycle, let's go now to the stages of AIDS. Okay. This is according to the World Health Organization classification. But before that, the main pathogenesis or pathophysia of your HIV is it preferentially infects and kills the CD4 T lymphocyte. That is the loss of a cell-mediated immunity. And if there is loss of the cell-mediated immunity, there is a high probability of opportunistic infections. Okay? The main immune response consists of a cytotoxic CD8 lymphocyte, but what happens in HIV, specifically it attacks the CD4 count. So, bababa ang CD4 count in HIV AIDS patient. Okay, these are the events that transpire from the primary HIV infection through the establishment of a chronic infection to the ultimate destruction of the immune system. We don't want to get into the ultimate destruction of the immune system. This means death. Okay? So, dapat sa primary HIV pa lang, na-recognize na natin yung mga patients natin. So, at first, there is a primary infection. There is establishment of infection in the lymphoid tissue. What happens is that there is massive viremia of virus in the blood with wide dissemination to lymphatic organs. It could be a partial immunologic control of the viral replication, or it could lead into trapping of virus and establishment of a chronic persistent infection. That includes fungal infection. No? These are the most common infections in HIV. Once there is trapping of virus, there would be immune activation that is mediated by the cytokines and HIV aberrant cell signaling, wherein there is an accelerated viral replication. If there is a rapid CD4 T cell turnover, the count for CD4 count will be decreased, and this one would lead to destruction of the immune system. So immunodeficient ang pasyenteng may HIV because it really destroyed the immune system of this patient, okay, leading to so many uncontrolled infection. Okay, this is just a summary of the stages of infection. We have primary, latency, and AIDS. Now, if you notice, these are the CD4 cells. Yan yung red na yan, red broken lines. During the primary infection, which will happen 2 to 4 weeks after exposure, look, after a 2 to 4 weeks infection, 2 to 4 weeks after contracting the virus, you have so many of the CD4 count. Okay, CD4 cells. Yan, no? Nakita nyo yung trend? Pababa. Okay, pababa ang trend ng CD4 cells until AIDS or death. Mababa na talaga. While these are the 8 anti-HIV antibody. Usually during this part, 2 to 4 weeks, wala pa masyadong detectable na antibody. But it will start, no, after 1 month, after 6 months, after 1 year, there will be an increase in anti-HIV antibody. But look, there will be a decrease, no, in an HIV antibodies. What is so much interesting about is the virus in the blood. During this primary infection of HIV, there is an elevated or there is a marked increase in virus in the blood during primary infection. And look, in latency or this stage of infection, the CD4 cell starts to decrease while your 8 anti-HIV antibody starts to increase because they've recognize that there is a need for antibody production. But look at the latency. There is virus in the blood which is almost undetectable. No, meaning you may screen a patient using antibodies because they are increased, but when you want to detect virus in the blood, it's undetectable using latency period. Okay, but the CD4 cell of this patient during latency stage starts to decline. Okay, that's so sad. The AIDS stage, there is this skin and mucous membrane immune defect, systemic immune deficiency. Totally, the HIV antibody is decreased also with the CD4 plus cell, but unfortunately, the virus or the HIV virus starts to increase until death of this person. Okay, so with this table alone, you may actually appreciate the primary infection. Now, what happens in primary infection? There is really starts to decline the CD4 cell, but there is a marked increase in viral load. Okay, in latency, there is increased antibody, but there is also an almost undetectable virus in the blood. Okay, and look at the time. It's usually 6 to 10 years, depends on your immune system. Ayan. So these are just clinical syndromes wherein you will be entertaining such clinical syndrome if you have CD4 count of less than 500, less than 200, and less than 50. I want you to all remember this. The CD4 count of less than 500, you may actually have, though HIV patient individuals may actually have a disseminated tuberculosis. Right now, if you are diagnosed with tuberculosis, infectious disease specialist now recommends HIV screening din if you are diagnosed with tuberculosis. What else? In HIV less than 500, HSV or herpes simplex virus esophagitis can develop, esophageal candidiasis, and Kaposi sarcoma. For less than 200 CD4 count, you may develop this type of pneumonia caused by Pneumocystis jirovecii, that is a Pneumocystis pneumonia, Toxoplasma gondii, producing a cerebral toxoplasmosis, meningioencephalitis by Cryptococcus neoformans, coccidioidomycosis. These are all mycologic or fungal infection. Okay. For less than 50, please take note of the three most common clinical syndrome if your patient has this CD4 count of less than 50. We have Mycobacterium avium, invasive pulmonary disease, Histoplasma capsulatum, producing histoplasmosis, and CMV is actually a cytomegalovirus retinitis. So please take note of that table. Okay, this is another illustration or table showing you the CD4 cell count that will start to decrease in acute HIV syndrome, but before ending it, before it goes to the clinical latency, it will go up high again and then eventually decrease na siya until death. As mentioned a while ago, the HIV RNA during acute HIV syndrome increase, but it will start to have some undetectable, but the level of it in AIDS patient is, starts to increase until death. Yeah, so this is again another table showing you the different stages of HIV/AIDS. Primary HIV infection, usually 2 to 4 weeks, and they are accompanied by flu-like symptoms such as fever, chills, night sweats, and rashes. But basically, before you consider an individual with HIV, make sure you ask or you ask your patient na yung medical history niya, kung nakipag sexual intercourse ba siya, etc. Sorry. Okay, going back, the CD4 cell in stage 1 is at least 500 cells per microliter. not applicable diagnostic test. During this time, the primary HIV infection, do not test yet. Now, it's too early. It's the timing of testing here, baka masabi mo false negative ka. So don't test during the primary HIV infection. In stage 2, that's your clinical asymptomatic or window period. This may last for an average of 5 to 10 years. This stage is free from symptoms, but your CD4 count will start to diminish or reduce to 350 to 499 per cubic meter. At this point, HIV antibodies are detectable in the blood. Okay. Next stage would be the symptomatic HIV infection. These symptoms are mild, such as skin rashes, fatigue, night sweat, mouth ulcers, fungal infection. At this point, the CD4 count still starts to decrease. And if you are in an HIV to AIDS stage, that's the most common. Opportunistic infections would include the pneumonia, the pneumocystis carinii or pneumocystis hirovecii pneumonia, the Mycobacterium avium complex, cytomegalovirus, toxoplasmosis, and candidiasis. Now look at this CD4 count in the HIV to AIDS stage. It's less than 200, or the percent of CD4 cells is less than 15% of all lymphocytes. So basically, if you have attained this cell count, CD4 cell count, at hindi pa nagpapacheck ng HIV, kung ayaw magpacheck ng isang pasyente ng HIV, ipa-check mo yung CD4 count. Because sometimes it's almost always you have an immune deficient virus. Okay? This is another, again, clinical stages as mentioned kanina. Usually asymptomatic with normal activity. In clinical stage 2, you have weight loss. You have a recurrent upper respiratory tract infection, or sometimes you have pneumonia. In clinical stage 3, more than 10% na yung weight loss mo. Meron ka ng unexplained fever, unexplained diarrhea, oral thrush, oral hairy leukoplakia, PTB in the past year, severe bacterial infection, or sometimes you are bedridden for less than 50% of the days in 1 month. For clinical stage 4, obviously, these are the AIDS-defining illness. And what are those? Usually, you may find Kaposi sarcoma, oral candidiasis in this stage already, in the clinical stage 3 and 4. Kaposi sarcoma or oral candidiasis, this one. You suspect HIV to this patient. Okay. Okay, as mentioned, in CD4 T lymphocytes, memory cells, and latency, the cardinal feature of your HIV, please take note, is the depletion of your T helper inducer lymphocyte or your T helper CD4 cells. Okay, these are the CD4 molecules. That's the major receptor for HIV. And the HIV co-receptor dun sa lymphocyte na yon is the CSCR4. That's your chemokine receptor 4. Okay. This is found in the host cell, CD4. Okay, what else? The other cells found in the lymphatic organs would be the monocyte and macrophages that will serve as the major reservoir for HIV in the body. Lymphatic organs would play an essential role in HIV infection, and that is actively replicating in those tissues. The microenvironment of the lymph node is ideal for its establishment and spread of the HIV. Now, cytokines are released that captivates a large pool of CD4 cell count that are highly susceptible to HIV infection. So, you would really want to suspect HIV if you have decreased number of CD4 count. Okay, kaya I have this patient. He doesn't want to be screened with HIV. So the other doctor, he has really high suspicion to HIV. So pina-CD4 count niya. And the results are very matching with the HIV. Okay, when you say AIDS or acquired immunodeficiency syndrome, this is the late stage of the HIV disease. They are characterized by less than 200 CD4 count. At this stage, there is the development of your opportunistic infections, selected tumors, wasting, and neurological conditions. These are yung may unexplained weight loss ka na, sobrang baksak na ng katawan mo, and other neuro conditions. Okay, what are the AIDS indicator conditions? 16% would really tell us that this one has AIDS if you have candidiasis, no, of the esophagus, trachea, bronchi, and lungs. Never mind this one. Pero look, they're all C, cytomegalovirus, cryptosporidiosis. These are all parasitic, viral, and fungal infections. But the most common of the AIDS indicator and conditions is this fungal infection called candidiasis. Okay, please take note. Please take note of this. Okay, so after discussing the different stages and life cycle of HIV, who should be really tested for HIV? Of course, person with high-risk behavior. These are the men who have sex with men, injection drug users, persons with multiple sex partners, persons who have exchanged money or drugs for sex, or that's transactional sex, persons who have had sexual contact with an HIV positive person before, or person at risk. No, you should be tested for HIV. Who else? Persons with certain medical conditions, such as these AIDS-defining illnesses that I mentioned kanina, with oral candidiasis, generalized unexplained lymphadenopathy, sometimes any STD would also mean an HIV diagnosis. And as mentioned, if you are recently diagnosed with tuberculosis, have yourself checked. Have an HIV screening then. Persons who have been sexually assaulted and persons who have had occupational exposure, such as those sexual workers. Those kailangan meron silang regular HIV screening. How do we diagnose HIV? Now, HIV infection established by detecting the antibodies to the virus, as well as viral antigens, viral DNA or RNA, or by viral culture. As mentioned, there are two types of HIV. The HIV accounts for nearly all cases, but for HIV2, they are found primarily in West Africa. It's less transmissible than HIV1. They have lower viral load, and they have a slower rate of CD4 decline. Okay, what are the standard serological tests? If we want to test or diagnose HIV, we screen the patient using ELISA test, or your enzyme-linked immunosorbent assay. That's a sensitive test for HIV or AIDS. Confirmatory testing is actually a sensitive. It has a 98% sensitivity and specificity. We use Western blot. Okay, so please take note of that. When we use ELISA, at least 2 reactive tests before we do a Western blot. And then the Western blot is always coupled with an enzyme immunoassay due to a false positive result, pero 2% lang. So again, before you proceed the confirmatory testing with Western blot, you should have at least 2 consecutive or 2 reactive tests with ELISA. Okay, because this is sensitive, most sensitive test for HIV. Okay, how do you interpret Western blot, yung diagnostic? Negative would be no bonds. If it is positive, usually your active is just a GP41 and GP120. These are your glycoprotein, your envelope genes, right? Your P24 and your GP120 or 160. These are bonds. So in this diagnosis of HIV, if it is positive, you've detected the GP41 and the P24. Okay, we can say that the Western blots test is indeterminate if there's presence of a bond pattern that does not meet the criteria of a positive result. Okay. Yeah, so this is an example of a Western blot. Usually have line 1, line 2. These are the positive control, the negative control, no? And negative, indeterminate, and positive. Okay, so can we interpret this? Yes. In the Western blot, you usually have the GP160 and 120. Those are your glycoproteins. Your P55, GP41, P31, P24. So in the Western blot, when we say positive control, dapat present to. Kaya nga positive eh. At kung negative siya, negative control dapat. So may mga 5 bonds yan. Now in sample A, kapag wala, negative. Kapag sample B, may mga bonds nga pero hindi naman. Wala naman siya GP116, 120. So this sample is indeterminate. But once sample C complied in every bonds, it is positive test. So Western blot is really for confirmatory testing. Okay. So anyway, this is the HIV viral structures. I've discussed this one by one. So please indicate what type of genes they are and what are the functions of each structure or viral structure in HIV. Diagnosis. Again, false negative results. This one actually occurs if tests were done during the window period. Doc, what is window period? This is the time delay from the infection to the presence of antibodies. That's why all individuals who are at risk of contracting HIV are advised to take HIV testing at least 1 or twice a year. Okay. So for example, you did something today, don't test tomorrow. Okay? Hindi pa magkakaroon ng positive or negative result. Obviously, it's negative, pero baka sabihin, ay, negative ako, kaya go lang. So usually, you have to wait for at least 3 to 6 months before doing the HIV test. And you'll have to repeat it after 6 months again. Okay? The antibodies are present within 6 months. Thus, testing of your HIV testing right away after a sexual intercourse is not recommended. Okay? Okay, how do we screen? No, usually counseling. The client-initiated or provider-initiated. When we say client-initiated, the client wants to be tested or screened with HIV, while your provider-initiated, usually your doctor. Now, pre-test, you do not actually get tested for HIV right away. There is a basic knowledge on counseling, risk assessment, positive and negative result interpretation, and then secure consent, either written or informed. And then the blood sample is taken from you. This one would ensure the confidentiality on the subject's identity. Okay. Now, if found twice positive for the screening test, you do the confirmatory test, which is your Western blot. If the first screening result is negative, no further test is done. That's why it is a sensitive test. The ELISA test is really a sensitive test. And then after which, post-test counseling will be done. Diba? Do not mind this slide. This is just an initial evaluation of doctors to an infected or HIV suspect. Napakadami. No, if you would notice, these are the routine care, no, blood count, RPR or the rapid the reagent test for syphilis, anti-toxoplasma for depression, for hepatitis, sobrang dami. Now, these are the tests done by the infectious disease specialist in an infected person. Okay. Okay. Now, when to initiate antiretroviral drugs? When we say antiretroviral drugs, these are medications not used for treatment, but not used to treat the HIV, but to decrease the chances of from HIV to AIDS. No, usually we give antiretroviral drugs to AIDS symptomatic for the CD4, which is less than 200 previously, and according to the World Health Organization, less than 350 ang CD4 count niya. Okay. HIV testing in the Philippines. There are 5 hospitals in our country that specializes with HIV counseling. We have the RITM in Alabang, San Lazaro Hospital in Manila, St. Luke's, Makati Med, and the Medical City. But right now, there are so many HIV testing hubs. No, when we say testing hubs, I think in Makati, Quezon City, Mandaluyong, near health centers, referral to these treatment hubs would offer. They offer screening tests, and then they are the ones. These are actually free. Yeah. And also, the antiretroviral drugs are free in the Philippines, especially in RITM and in San Lazaro. You should have a regular checkup lab. Okay, these are the antiretroviral drugs given to that are being given to the patients here in the Philippines. Pero don't memorize this. Nah? This is just something that I want you to show that there are medications for HIV, but not totally to treat HIV. Okay, it will just decrease its progression to AIDS. Okay, what about vaccines, Doc? They are safe and effective vaccine for HIV. Now that offers best hope, but there are no vaccine right now for HIV. It's very difficult to produce or develop. Why? Number one, the HIV mutates rapidly. Number two, they are not expressed in all cells that are infected, and they are not completely cleared by host response after a primary infection. Diba sabi natin, after primary infection, it goes to a latency. Matutulog lang siya, but you have the virus. Okay? You're not symptomatic, you don't have symptoms, but you are really infected with it. All right, so the following vaccines are recommended for all people with HIV. So ano-ano yung mga vaccines yan? You may actually recommended are hepatitis B, your flu vaccine, your human papilloma virus, your pneumococcal, your DPT, and the COVID vaccine. They are all recommended for all people with HIV. All right, so question, what is the current rate of HIV diagnosed in the Philippines? Actually, this is outdated. It is now 1 new case per hour. Okay. What city in the Philippines has the greatest prevalence of HIV among men having sex with men? Can you guess? Can you write it in the chat box? The answer is Cebu City. Okay. I think after Cebu, it's Quezon City and Manila. And Davao, I think wala pa siya sa top 5. Nasa top 10 pa siya. Pero they say the most the city with the most number of HIV diagnoses per day is Cebu City. All right, so let's talk about other virus in the Retroviridae family. We have this. No? There are 7 genera, but we will just be talking about the Delta retrovirus. That's your human T lymphotropic virus or HTLV, or the bovine leukemia virus, no? So the Delta retrovirus. The HTLV1 is the causative agent of APL. That's your adult T-cell leukemia lymphoma and tropical spastic paraparesis. It does not carry an oncogene, meaning they are not cancer-causing. Your HTLV2 naman has been isolated but has not been conclusively associated with any specific disease. But either or, your HTLV1 and 2, they share about 65% sequence homology and they will display significant serologic cross-reactivity. But HTLV1 is this one, much more not common, but it will cause disease but does not carry oncogene. Retroviruses are trans-regulating. They carry a gene, they taxed it, whose product will alter the expression of other viral genes. So, sabi natin, they don't carry an oncogene, right? But they carry a certain gene, the trans-regulating gene. The trans-activating regulating genes are necessary for the viral replication of this HTLV, or human T lymphotropic virus. They may contribute to oncogenesis by also modulating cellular genes that regulate cell growth. Okay, it actually are distributed worldwide. No, the HTLV. It has 10 to 20 million infected individuals. Clusters of this HTLV associated disease are found in certain geographic areas such as in Japan, Melanesia, Caribbean, Central and South America, and parts of Africa. But it's so sad to say, not just the HTLV, the HIV diagnosis starts to decline worldwide, but in the Philippines, it's still uprising. That's so sad. The transmission of this HTLV, human T lymphotrophic virus, involves cell-associated virus, 15 to 25%, mostly from mother to child transmission, just like your HIV, blood transfusion, sharing of blood-contaminated needles, and sexual intercourse. HTLV1, no, they are linked infection with the HTLV1 called the associated myelopathy or tropic spastic paraparesis. The primary clinical feature of this HTLV1 is really progressive weakness of the leg. So parang kang paralyzed. Kaya nga may isa silang infection called spastic paraparesis. The patient's mental faculties or mental condition remains intact, but this one, the HAM and the TSP, is described as being of the same magnitude and importance in the tropics as is multiple sclerosis in Western countries. But this is not common anymore. Okay? The HAM and the TSP. That's it. That ends the HIV/AIDS and other retroviruses lecture. Thank you for listening.